Today, I will discuss how bioinformatics methods help us decode the molecular mechanisms underlying traditional Chinese medicine. Traditional Chinese medicine, or TCM, is a rich and complex medical system with thousands of years of clinical use. However, there are fundamental differences between traditional Chinese medical concepts and modern pharmacology, which greatly hinder the modern development of TCM. Therefore, I will be focusing on how bioinformatics provides the tools to systematically analyze that complexity of TCM in this presentation. From a modern biological perspective, TCM is an extremely complex system. Most of the Chinese medicine prescriptions contain complex combination of herbs, each composed of many chemical compounds. And each compound might interact with multiple proteins, which then influence multiple biological pathways. This creates a many-to-many relationship that is very difficult to untangle using traditional experimental intuition alone. So the key question becomes, can conventional pharmacological approaches handle this level of complexity? And their answer is there are limitations of the traditional reduction approach. Conventional drug discovery often relies on one drug, one target model, but TCM effects typically arise from synergistic interactions across many targets and pathways, not isolated molecular actions. This mismatch between the methods we're using and problem explains why early mechanistic studies often produce fragmented or say incomplete explanations. And because of this limitation, researchers began to look for a framework that could actually handle interactions rather than the isolate compounds, or say components. And system biology exactly brings us a new perspective. System biology is a biology-based interdisciplinary field, and it offers a way to view biological processes as an interconnected system rather than isolated parts. Instead of focusing on individual molecules, system biology emphasizes interaction among genes, proteins, and pathways. However, this perspective only becomes practical when those interactions can be represented and analyzed computationally, which is exactly where bioinformatics and network pharmacology enter. Network pharmacology translates system biology into computable framework. In network pharmacology, compounds, proteins, targets, and biological pathways are represented as nodes connected by edges. This allows us to study properties such as highly connected targets, pathway clustering, and potential synergistic effects. But here comes the bottleneck. A major bottleneck in TCM analysis is that most herbal compounds have unknown molecular targets. Unlike the approved Western drugs, the majority of TCM compounds lack experimentally validated protein targets. And without knowing these targets, we cannot construct meaningful networks, and the entire computational framework essentially doesn't work, or say it collapses. And this bottleneck motivates the use of computational target prediction, which reframes this limitation as a computational inference problem. Bioinformatics approaches assume that chemically similar compounds tend to interact with similar biological targets. By comparing unknown TCM compounds to known drug molecules, algorithm can generate ranked lists of likely protein targets. And one widely used example of such approach is Batman TCM. It is a bioinformatics analysis tool for molecular mechanism of TCM that implements similarity-based target prediction within a bioinformatics framework. It interacts chemical similarity measures with known drug-target interactions to infer potential targets for TCM compounds. The method integrates multiple types of similarity information, including chemical structure similarity between compounds and biological similarity between targets, and combines them into a unified scoring scheme. And rather than producing a single prediction, Batman TCM outputs a ranked list of candidate targets for each compound, allowing flexible downstream analysis. And And something that is very important to note is that it also supports ab initio analysis, meaning that the targets can be inferred even for compounds with no prior biological annotation, relying solely on their chemical features. This makes the framework particularly suitable for TCM, where novel and poorly characterized compounds are very, very common. However, prediction alone is not enough, right? We must ask how reliable these predictions are. Reliable target prediction must first be statistically validated and then translated into biological meaning through data-driven interpretation. And in Batman TCM, the reliability of predicted compound-target interaction is evaluated using cross-validation strategies and performance metrics such as ROC curves and AUC values. High AUC scores indicates that a model can effectively distinguish true interactions from false ones, providing confidence that the predicted targets are not random. And as we can see in the graph right here, the AUC values are all relatively quite high, nearly to 1. So we're quite confident about the results. However, statistical accuracy alone does not explain what these targets collectively represent biologically. And to address this, bioinformatics applies functional and pathway enrichment analysis, such as gene ontology and KUGG analysis, to identify biological processes that are statistically overrepresented among the predicted targets relative to a background model. By focusing on patterns across many targets rather than individual proteins, this approach ensures that biological interpretation is driven by statistical significance and reproducible data rather than selective or, say, intuitive reasoning. But of course, the limitations still remain. Current challenges are largely computational in nature. For example, incomplete databases, uncertainty in prediction, and simplifying model assumptions remain to be very important issues. Future advances such as machine learning and multi-omics integration are expected to be refine these models and further refine these models and improve accuracy. In summary, bioinformatics provides a structured and data-driven framework for studying traditional Chinese medicine as a complex biological system. And this presentation has shown how bioinformatics methods, including target prediction, statistical validation, network analysis, and enrichment-based interpretation, can be combined into a coherent and analytical pipeline for TCM research. By modeling TCM mechanisms as molecular networks and interpreting them using statistically grounded approaches, bioinformatics enable mechanistic insights that are reproducible and transparent, which enables the modernization of TCM. And that's actually the end for this presentation, and thank you so much for listening.